Okay, so welcome for the last part of the lecture. I told you so far that in population genomics, and when we look at selection in genomes or populations, we only look at one thing, which is allele frequencies. Well, now that's some cases where actually we don't really look at that, or we look at this plus extra information on the phenotype or environment. So, for example, if we are searching for loci or genes that are responsible for local adaptation, let's say, then one way to do it, which is rather old, is using QTL approaches. So, QTL approaches requires, first of all, two different parents. One that will be have a phenotype A and the other phenotype B. In our case, we are talking about adaptation. And then from these parents we will need to create a progeny, which is then F1, which will be heterozygous, parent A, parent B. Then from the F2s on, you're going to start to have this mosaic pattern simply because of recombination. Like recombination will make that these two chromosomes will recombine and then start to create all this mosaic that you see here. Once we have this, then we can start to work. And what we do is we do phenotype, and we assess whether these F2s, each of these individuals, are adapted or not adapted, let's say, and we sequence their genome. For example, here, imagine that among them, 4 of them show some sign of adaptation, and 1 of them does not. Then, because we sequence and we know that, okay, this is rather coming from this parent, this comes from the locally adapted parent, and so on and so forth, we are able to narrow down the regions that could be responsible for adaptation. We simply do an association. And for example, here, the candidate region could be here, this one, between these two red lines, simply because that's the only correlation that we can find where adapted individual shows a piece of the genome from the locally adapted parent, while the non-adapted F2 individual doesn't show, or show rather the allele from the non-adapted parents. So this, we have a perfect correlation between the genotype at this locus and the phenotype, adapted versus non-adapted. And that's how we determine a QTL. So here's an example here. So this is the example of a fungi that affects wheat, for example, and people notice that depending on the region that wheat is grown, this fungi is able or not to affect wheat. And then for that, they then did a QTL analysis on this fungi for the trait which is temperature sensitivity, because they assume that the authors assume that this fungi has some temperature sensitivity, which could explain why it could be found on some regions on wheat and others not. So they did that, so they created lines of different fungi, and look whether they could find temperature sensitivity QTLs responsible for this. So that's how it looks like. So this is all the chromosome of this fungi, and what you have here, this is called lot score. This is showing the association, how strong is the association between the phenotype that you measure and the genotype at the specific locus. So stronger, the more probably, or the more likely it is that these locus contain the regions or the gene for temperature sensitivity. And then in this case, then we establish a lot score threshold, which is based on permutation tests and everything. We will do that in the practical, hopefully. And they could find 3 or even 4 QTLs that could be responsible for temperature sensitivities. So again, these QTLs show association between genotype and phenotype. Okay, but the thing is, we cannot always do F1s, controlled F1, controlled F2s between parents with specific traits. And of course, this is the case of humans, and as you can imagine, we are not really allowed to produce controlled F1s and F2s of different ethnicities or stuff like that. So instead, an alternative to QTL is called GWAS, which means genome-wide association studies. And this was done here. This is a review of all the GWAS that has been done in humans so far for different traits and for local adaptation, actually. So, for example, local adaptation to fish diet on northern hemisphere, so which means some adaptation of metabolism, or adaptation to specific or to UV exposure, like in Africa, and stuff like that, or lactose intolerance, these kind of things. So these are the kind of traits for locally adapted populations that we are looking at. And instead of then measuring or sequencing F2s or F1s or whatever, instead we're going to sequence the whole population. So we're going to sequence populations that are non-adapted to whatever we're looking at, or that are locally adapted. Once we have this, we sequence them, as I said, and then we can find the specific mutations that are discriminating non-adapted versus locally adapted populations. This is what you have here. This is a probability that this or this or this mutation, for example, discriminates non-adapted versus locally adapted populations. So again, in the end, it's the same idea. So you do a genotype-phenotype association, except that you group. So you know a priori phenotype, so locally adapted, you group them, you sequence them, and you obtain genotyping. It has a better resolution than QTLs in general. Like, if it works well, you can basically find a single SNP, so SNP for single nucleotide polymorphism. You can find the effect of a single SNP on the phenotype, rather than regions, large regions, which are harder to find or to associate functional with a phenotype. Of course, there are some drawbacks, and one of them, when you select these groups you want to sequence, you need to know about the demography and especially the population structure. That can influence a lot the discrimination between two different populations, as you can imagine. So this is an example for skin and eye color, for example, for European versus African descent humans. And these are typical genes involved in light perception, and this one involved in skin pigmentation, which shows again a strong association between then local adaptation versus genotype. And again, you see that this is a single gene can be found. And what you see here, like long peak, sort of, is again due to genetic linkage. If you remember, then probably this is a mutation that is responsible for skin color, let's say, because it has the highest association, and all the others are just alleles or mutations around this mutation. And because there is genetic linkage between them all, then they will always find to be associated with the phenotype, even though this one is the one which is responsible. So keep in mind this, and it's the same with QTLs. Genetic linkage will always gather mutations that are next to each other, even though it could be that only one of them is responsible for the phenotype. Now, if we can do another thing, so especially with plants, with humans it's a bit more complicated because they move, but for plants we can do environmental association. So here, instead of having a trait, we will have an environment. We're going to have plants, okay, humans pictures here, but it doesn't matter. So plants growing in location 1, plants growing in location 2, and another plant growing in location n. And each of them will have each of these locations. Now there is plenty of database of climate, so each of these locations will have a specific climate. And then once we have that, we can simply do again an association, this time between genotype or SNPs and climate variable. This is the case here, for example, a study that was made some years ago in Arabidopsis thaliana, and they associated temperature seasonality, which means difference between summer and winter, and then specific SNPs. And what they found there, for example, so like in regions where there is a high temperature seasonality, there is a specific allele of this gene, which is an epigenetic regulator, which is found. And that's how the map looks like. So you have either a functional allele or a stop codon allele, which is either blue or red. And what is pretty clear is that the functional allele is most of the time found in rather temperate regions, so then the temperature seasonality is rather low. But if you go for more extreme climates with very cold and very hot, very cold winter, very hot summer, actually the majority of the alleles that are found are rather these stop codon alleles. And again, now that they found this allele, if they plot the temperature seasonality versus the type of allele they have, either stop codon or functional, they do find this relationship. So like accessions with living in higher temperature seasonality most of the time have this stop codon and vice versa. So this is one other way to do. Yeah, and that's it. So in general, as a conclusion for the lecture, so remember that neutral molecular evolution is a null hypothesis. Like selection is really explaining like a tiny part of the genetic diversity that we can observe. And demography as well can have large effects on molecular evolution. And because of that, then we need to test always demographic effects before we can even search or start searching for selection. So I showed you many methods, but many of them actually rely on a similar approach. When we search for selection, we always compare divergence between lineage versus variation within a lineage, and that tells us a lot. Of course, I also showed you then GWAS and QTLs and environmental associations, and that depends a lot on your model study. Basically, if you have animals, environmental association is probably not the right thing to have, and so on and so forth. Thank you.